Guess what? This is one of our winter shorts. Yep, that's right. It's a shorter than usual episode in which I present one or two, or maybe three or four classic evergreen segments from previous episodes that is remastered, reconstituted, and recycled for your listening and learning pleasure. But mainly it's to give me a break for self-care over the holiday season. We'll be back to our regular programming in late January.
Welcome to the A&P Professor, a few minutes to focus on teaching human anatomy and physiology with a veteran educator and teaching mentor, your host, Kevin Patton.
In this episode, I discuss the temperature of mitochondria, the immune role of platelets, the odd shape of the Golgi Apparatus, and green pens for grading. Today's lucky number is 10. Why 10? Well, it turns out that there was some information just published that suggests that it's quite possible that the temperature difference between mitochondria and the rest of the cell, the rest of the body, is about 10 degrees Celsius. So 10 for 10 degrees. That's a little bit of information that isn't central to our understanding of human anatomy and physiology, but we certainly talk about the fact that mitochondria do produce heat, and a lot of it. And that helps us explain thermoregulation in the body. It helps us explain how muscles, when their mitochondria, which they have bajillions of, are really cranking, that's going to possibly overheat the body. And we're going to have to turn on our cooling system to get it to cool down, or take various behavioral strategies to cool things down. And when we're too cold, we can start shivering our muscles and doing other muscular activity to kick those mitochondria in gear and get our body temperature warmer than it was before. 10, a little number we can stick in our pocket and pull out sometime when we're telling the story of metabolism or thermoregulation or homeostasis or who knows what. Platelets. We all know about platelets, right? There's billions of these tiny little cell fragments, something like 750 billion of them circulating around in the human bloodstream. And we know that they have broken off from a much larger body, a cell called a megakaryocyte. And as they travel around the bloodstream, when an injury occurs in a blood vessel, the collagen in the vessel wall is exposed, and that causes some of the platelets to become sticky. And as they stick to the collagen and start piling up, they form a platelet plug, which helps stem the flow of blood, but it also, along with some other chemical pathways, triggers a whole cascade of reactions that eventually result in a blood clot. Now, that's the story we all tell in our class, right? But did you know that they have other helpful jobs too? One of those that was recently outlined in the literature is that they can round up bacteria and literally feed them to our immune cells, which in turn can devour the bacteria and help make us safe. So we can say that platelets, besides having a role in hemostasis, have an innate immune function. What are these functions that have been recently outlined? Well, to just boil it down to a few simple points, one thing we can say is that when you have a vessel injury or inflammation, platelets that contact the intact collagen, they stick together. Those are the sticky platelets we all know about. But we now know that platelets that do not contact collagen are motile. They can move around. And those motile platelets, they change shape. The platelets to start out with look kind of like a fried egg when you look at them under an electron microscope. They're kind of flattened out and have a little bulge in them. And they're going to change when they're motile and become sort of an uneven half-moon shape. And it turns out that half-moon shape helps them better navigate the strong shearing forces that exist in the flowing blood. And not only that, but these motile platelets, they can even navigate upstream. That is, they can go against the flow of blood. Now, that's something I didn't expect to see, but, well, we're always finding new and amazing things about how our body works. Another thing that the researchers found is that these modal platelets, they use mechanical force, that is the force of the molecular motors in their cytoskeleton, to pull particles, including bacteria, from the surrounding substrate. So if there's some bacteria piled up on a vessel wall at the site of an injury where there's some inflammation going on, those modal bacteria can go over and grab a hold of those bacteria and pull them off the substrate. And then they collect those bacteria and actually form little bundles of bacteria. So they're gathering up these potentially harmful bacteria, they're bundling them up, and that in turn is going to facilitate neutrophil activation. And we know what that means, right? That means that we're going to have some phagocytosis going on. So the immune cells, the neutrophils, are now going to be able to take those bundles of bacteria from the platelets and gobble them up. So that makes the whole process of what the neutrophils are doing in an inflammation response that much more efficient, something that we didn't really know about before. Now, I'm not saying that we need to incorporate this whole story into our course, but it might be a good thing to mention when we're talking about platelets that, hey, here's their main job, but they do other things too. For example, let's put it in there. Just drop it in, simplify it, don't make a big deal out of it. And then usually a little bit later is when we run across a discussion at some level of innate immunity, and we might want to drop it in there and mention it like, well, we're going to cover some of the main mechanisms of innate immunity, but there's lots of other things going on in terms of immune protection. Remember those platelets we talked about and how I mentioned that they can gather up bacteria, the ones that aren't being involved in a platelet plug, they can gather up the bacteria and help make that phagocytosis by the white blood cells that much more efficient. So a couple ways you can drop it into your course if you want to. Even if you don't, boy, it's just an interesting story to hear about, right? And there are links to more detailed information in the show notes and also in the episode webpage. I know. Nobody is eager for a sponsor message. But not only do federal regulations require it, believe it or not, our sponsors expect it. And they're financially supporting this podcast so that there's no cost to you. Yay! Besides that, I'd be mentioning them from time to time anyway, because all three are great organizations that I'm intimately involved with, and they benefit all faculty teaching anatomy and physiology. Can I say that? That I'm intimately involved? I hope that's okay. One of our sponsors is AAA, the American Association for Anatomy. Check out all the resources and membership information at anatomy.org. Another sponsor is the Master of Science in Human Anatomy and Physiology Instruction, the HAPI degree. Check out this online graduate program at Northeast College of Health Sciences at northeastcollege.edu/HAPI, that's HAPI. And rounding out our sponsor list is HAPS, the Human Anatomy and Physiology Society. You can check out resources and membership information online at theapprofessor.org/HAPS, that's HAPS. Did you ever wonder why the Golgi apparatus looks so odd compared to other membranous organelles of the cell? I mean, really, wouldn't you think that the forces causing the other membrane-bound structures to form more of a globular shape would also cause the sacs of the Golgi apparatus to be more, well, round? A number of years ago, the journal Cell published an article that answers that question. And it revealed a really cool mechanism resulting from the primary function of the Golgi apparatus. As we know, the Golgi apparatus processes and packages proteins that arrive from the endoplasmic reticulum, or ER, by way of the ER vesicles. The central structure of the organelle is the Golgi stack, or dictyosome, which sort of resembles a stack of hollow pancakes that are kind of curved around a little bit. Golgi vesicles pinch off of the first sac, or cisterna, which is called the cis face. And then the vesicles move to the next cisterna, and then the next, and finally to the final cisterna, which is called the transface. Then a vesicle pinches off the cisterna at the transface and moves toward the plasma membrane, where it fuses and releases or secretes its contents into the outside of the cell in the process called exocytosis. Okay, so we know all that. The new data suggests that the budding of vesicles and their movement toward the plasma membrane rely on the function of a protein called GOLPH3 or GOLF3. This tiny protein connects special phospholipid molecules in the Golgi membrane to myosin molecules. It's a kind of myosin called MYO18A. This myosin, in turn, is attached to F-actin filaments of the cytoskeleton. Okay, so let me go through that again so we can picture it. This is hard on a podcast when you can't draw a picture. Okay, so what I'm saying is it's all about a protein called GOLF3. This tiny protein connects special phospholipid molecules in the Golgi membrane to myosin molecules. These myosin molecules in turn attach to the F-actin filaments of the cytoskeleton that surround the Golgi apparatus. Well, you know what that means, right? Myosin and actin connected? Yep, you guessed it. The myosin is a motor molecule that pulls the attached Golgi membrane along the F-actin filament. So it stretches out and away from the center of the Golgi apparatus. And as it stretches, it takes on its familiar elongated shape. And then thwap. Yes, that's thwap. THWAP. I guess that's how you spell it. I don't know. Then thwap. A vesicle pinches off and is carried away. The short version of this story is the Golgi membranes flatten out because they're being pulled outward by the cytoskeleton in a process that produces the budding of vesicles. It's as simple as that. So now when you're describing this amazing little organelle in your A&P class, you have a new little twist to add to the story. And you probably already know it's named for its discoverer, Camille Golgi, who first observed it in 1897. So remember that date in case you're ever on Jeopardy! Or maybe this date's better. In 1898 is when he published it. And when he published it, a lot of scientists thought he was wrong. They thought that he was using this crazy Golgi staining process and it was creating an illusion of there being this organelle inside the cell. And because, before then they didn't really know about organelles. But then in the 20th century, when we start using electron microscopes, we finally proved that Golgi was right all along. By the way, here's another little interesting bit of trivia, and maybe it'll show up on Jeopardy, you never know, or at your next trivia night. Golgi complex, or its synonym Golgi apparatus, are among the very few eponyms that appear in the Terminologia Histologica, or TH. You probably already know the TH is the, quote, official list of microscopic anatomy produced by the FIPAT, that is the Federative International Program on Anatomical Terminologies. And the TH and other official lists, they tend to replace eponyms with more descriptive terms. So yay for Golgi, his name lives on, even in the TH. Okay, here's a really little thing. But I think it can have a big impact on the effect of our teaching on students. And that is, what color pen do you use when you're grading papers and marking comments on papers and lab reports and so on, when you're sort of sketching out things in the margin of a student's notebook to help explain an idea? If you're like me, who began teaching during the dawn of humanity, we were all issued red pens, and that's what we expect from teachers, right, is using a red pen. It stands out from whatever blue or black or whatever color pencil marks that they're making on their paper, so it's very clear who wrote what, and the comments really do pop out. The problem is that our students have, lived part of their life already spending decades being corrected by teachers in a red pen. And some of them have had some not so good experiences. And not only that, but psychologists tell us that the color red is an alarming color. There's a reason why fire alarms and even fire trucks and so on are colored in red, because it has a certain impact on our psyche. Psychologists also tell us that the color green has a more positive impact on our psyche. It's the color of nature. It's a very peaceful color. And so it's got to be more than 10 years, maybe 20 years now, I have avoided using a red pen, and I almost entirely use a green pen. If you run into me at a HAPS conference or somewhere else sometime, tap me on the shoulder and say, hey, can I borrow your pen for a minute? And I will pull out a green pen, probably a felt tip. And that's because that's my go-to color now. It stands out from what color other people are using, especially my students. And so when I'm putting grades or comments on papers, I do it in green. I think that it has a little bit of a positive impact, and all those little positives that we can do in our teaching, I think add up and add to the kind of atmosphere that we want to build in our course, that is an atmosphere of support and progress, and not an alarming red kind of course. If you don't see links in your podcast player, go to the show notes at the episode page listed at theapprofessor.org/podcast. And while you're there, you can claim your digital credential for listening to this episode. And you're always encouraged to call in with your questions, comments, and ideas at the podcast hotline. That's 1-833-LION-DEN or 1-833-546-6336, or send a recording or written message to podcast@theapprofessor.org. We'll pivot back to fresh, full episodes in late January, when I'll be giving my predictions for next year. If you have a prediction for where A&P instruction is headed, please send it in. I'll see you down the road.
The A&P Professor is hosted by Dr. Kevin Patton, an award-winning professor and textbook author in human anatomy and physiology.
This episode has been reconstituted from concentrate using only 100% pure electrons from a natural spring.